So over to Chris Quince, who is a group leader at the Earlham Institute. And Chris has been very heavily involved in the UK wastewater sequencing project to look to see what's going on in wastewater with respect to COVID-19. And before that, he's done many different things on microbiome studies and metagenomics. So Chris, I won't use any more of your time. Over to you. Thanks for coming.
Oh, yeah, no problem, Nick. Thanks a lot. I'll start sharing my screen then. Right. Okay. So you're seeing those slides, all right? Yeah. And Nick, if it gets close to 3 o'clock, say something, because I do have to go off to another meeting, but I haven't got that many slides.
Okay.
So yeah, hopefully we'll get it through it. Yeah, so as Nick said, I've been quite involved in assisting the UK in their wastewater COVID monitoring project, the DHSC. Yeah, my role has been sort of designing some additional bioinformatics tools that might be useful to them. So I'll talk a bit about those tools and give you a sort of more general introduction to COVID wastewater sequencing, although it's sort of becoming a sort of pretty well-established thing now. I suppose the big appeal of it is it's a way to cheaply and quickly monitor COVID in an entire population, and it doesn't. You can observe lineages even if people aren't reporting for testing, and potentially it could act as an early warning system, particularly in places like developing countries where you haven't got a great capacity for testing available. Now COVID is in the wastewater. Well, at least at the time I put this together, there was no evidence that any of that sequence is actually infectious. And in fact, I think there's increasing evidence that COVID doesn't remain infectious in the environment for very long at all. But the key thing is you've got that sequence from a mixture of populations that you can then use to monitor the entire the entire population. But the bioinformatics has to be somewhat different, and that's my involvement. And that's because you have this mixture of genomes. So you can't just run the standard tools, which are assuming that a single genome is present there. Now, it's now sort of established in a number of countries. So as I say, my experience is with the UK, where they're monitoring a large fraction of the population, actually on a daily basis. So there's a lot of data being generated. I personally don't get to see any of it. As I said, my role is to assist in the development of programs that are being run at the JBC. So as I said, there's this mixture of lineages present. Now currently the DHSC in the UK is just looking at SNPs and using that as a way to get an idea of what might be in a sample. I was aiming to develop a pipeline that would enable them to automate that process. I started this, I think, in May of last year. At that point, nothing was really available as an alternative. There are now a few pipelines that can do this. One is based on a tool from metagenomics for read mapping with ambiguity called Callisto. Another one is lineage spot, which is looking for unique variants in different genomes. We took a somewhat different approach, as I'll outline. Now, it is though actually a rather difficult problem. So why is it difficult? Well, one of the fundamental difficulties comes from the fact that COVID lineages share variants. So there's very few SNPs that are unique to a variant. Most SNPs are shared across multiple variants, and it's not even something that has, a very clear phylogenetic signal. Those SNPs are, I guess, through recombination, they're being shared throughout the tree. So when you have a sample that can be a mixture of multiple variants, then there's a lot of ambiguity, because, you know, basically, if you've got two lineages present, you can probably make a third lineage up that isn't present out of mixtures of SNPs of the ones that are, basically. So that's why it's a challenging problem. It's also somewhat challenging because of the tiled nature of the amplicons. Again, this is only important when we're thinking about mixtures, because, you know, if you've got a sample, you know all those amplicons are coming from the same genome. But yeah, with a mixture of samples, you have to take into account the independent PCRs on each of those amplicons, and those PCRs generate noise. So obviously there's sequencing noise, which may be quite important on if you're using nanopore. All our analysis is based on Illumina, where the sequencing noise is almost irrelevant, but the PCR noise can be very significant, as I'm showing in this plot here. Here I'm actually showing the frequency distribution of PCR errors that we've calculated on some synthetic samples at different levels of edit distance to the true sequence. So you can see that even at relatively high edit distance, so 4 changes compared to the true sequence, you're still finding PCR errors with, let us say, 0.2, i.e. 20% of the sample. So you're generating these relatively high frequency errors that can then appear like true variants. So yeah, so the aim is to reconstruct from the sequences on the different amplicons. You're aiming to reconstruct the most likely combination of lineages in your sample and their proportions in that sample. So how do we do that? Well, we've developed a pipeline that we call CovMix. It's slightly different to the SNP-based approaches, because we basically do everything with amplicon variants rather than SNPs. So we start with a database of genomes. We use a set of primers, could be Arctic, could be Nijmegen, to fish out the amplicons of the reference sequences. And then we basically map the reads against the Wuhan reference to get their location, remove primers, but then we overlap reads matching to the same amplicon, and it's those merged amplicon reads basically that are the input to our algorithm. Yeah, so it's implemented in SnakeMake. And yeah, as an example, I've used a database of 1,200 or so lineage genomes from GISAID. And yeah, you can, currently we can support Arctic version 3 and both versions of the Nijmegen primers. So how does the algorithm work? Well, it's a Bayesian algorithm, although it's technically an approximate Bayesian algorithm using something called variational inference. But that's rather similar to an expectation maximization algorithm. The basic principle is you iterate between these steps. We assign the reads to the reference amplicons. We have an estimate of error rates on those amplicons, which we update each iteration. And then given the known mapping of genomes to variants, we assign the reads across the possible genomes which are consistent with them. And from those latent variables, we're then able to actually update the mixture proportions. All right, so I'm going to present a little bit of synthetic data that we've applied the pipeline to. This synthetic data was generated by Terry Burke at Sheffield and then sequenced in Liverpool by Steve Patterson's group. The synthetic mixture consisted of 2 twist control genomes. So you can buy known COVID genomes from twist. They are in parts, which is something we have to try to take account of. And Terry mixed those up. He got 2 genomes. One I'll call MILK and the other one I'll call SHEF. I've got the exact names of those lineages later on in the talk. And they were mixed in proportions of 0% MILK, 100% SHEF to the reverse. Yeah, 100% milk, 0% SHEF, and then multiple combinations in between. What I'm showing in this plot is just the frequencies of reads we got on each of the amplicons. So there are some amplicons that drop out, and that is probably because that's at a breakpoint in the twist genome, and some amplicons are better represented than others. But at least for the high concentration samples, so samples were generated in 2 configurations using a low amount of input and a high amount of input. And I'm actually only going to talk about the high concentration. Well, I've got a few results from the low concentrations, but you can see we've got decent reads across amplicons for the high concentrations, less so for the low concentrations. All right, so we used CovMix to profile these samples with as I said, the GISAID database and Arctic version 3 amplicons. So the first question is, do we get the approximately the correct proportions of the 2 actual genomes that are in there? And we do for the high concentration samples. And so on the left here, I'm showing the actual versus predicted proportion of the milk genome. And we've got a good correlation for the high concentration samples. For the low concentration, I mean, there is some sort of correlation, but there's a great deal of noise. We do somewhat less well for the Sheffield genome, as we still have about 20% prediction of other genomes, even when that should be making up 100% of the sample. But the correlation is good. We do, though, get quite a lot of false positives. So this is what I'm showing here for a sample. This was 50% of one genome, 50% of the other. So we do predict those 2 genomes correctly, and in roughly 50% proportions. But we've got a sort of long tail of low frequency genomes. And what is happening here is there are PCR errors. So if you have a PCR error that turns one of those true amplicons into something else in your database, then the program is predicting a relatively small abundance across the sample of that as a false positive. And on this basis, we think the sort of reliable detection limit of the algorithm is around 2.5%. You can actually get a sense of what is a false positive by looking at the by breaking down the predictions by amplicon. So this is what I'm showing here. So now I've got each of the lineages, and across amplicons, I'm showing the proportion of amplicons that were assigned to that lineage. So you can see the sort of topmost abundant false positive, B1434, is basically, there's one amplicon where an error has made, has made the observed reads look like the incorrect variant rather than what's really there. So we find this very useful, looking at the breakdown across amplicons as a way to sort of further interrogate the data. Another thing you can do is look at your lineages across the phylogenetic tree. That's what I'm showing here. And this illustrates what I was saying at the start of the talk. So the false positives aren't all around as a sort of cloud around the true sequence. They're distributed through the tree. And that comes down to this, this recombination of variants across lineages, and hence why an error can then give you these false positives that are relatively phylogenetically distant from what is really there. One thing we can do to improve the results is filter the database a bit. One of the reasons this is so challenging is because we're using all 1,200 reference genomes at this point. If we sort of subselect references distributed through the tree, and then at the same time also select amplicons which are well represented across those references. Yeah, because another source of uncertainty is when from the genome sequence you have errors in an amplicon, so we can't use it in our database. And then you sort of, you're likely to pull in incorrect predictions on that amplicon. So you can sort of prune your database to improve the results. And that's what I've done here. I've made a set of 191 sequences and just 89 of the 98 amplicons. And yeah, then you can see that our overall predictions are getting rather better, and we've got less false positives overall. That's what I'm showing here. We've over across the whole data set then of the high concentration samples, we only ended up with 16 or so false positives. All right, so just to conclude then, I'm going to present a little bit of real results where the JBC, as you've been using our program, as I said, to support their surveillance efforts just based on SNPs. So these were some samples which I think were taken from quite early last year, from London. Oh, actually, I shouldn't say that. Yeah, so quite early last year. And yeah, I mean, this made sense. These predictions made sense in the context of the epidemic at that point. Just last December, things got interesting, obviously, with the appearance of Omicron. And actually, in early December, CovMix was used to process a set of 8 samples taken from a sewer network. So that this is where they've got they've taken grab samples, so a single sample from a set of locations in the same area, and Hugh Bear ran that on CovMix against just 10 or so lineages. And yeah, this was he was actually able to detect significant proportions of Omicron. Yeah, almost as soon as we knew that it might be in the UK from the wastewater sequencing, as well as the other lineages which were around at that point. And you can see this is interesting because there's a fairly complex mixture. And we did the same thing again, breaking it down by amplicons to see whether these some of these might be false positives. And yeah, the Omicron in particular is being picked up with yeah, a great deal of certainty. And yeah, since then, Hugh Bear's improved his reference sequences to fill in some of the gaps in the amplicons. At this point, Omicron wasn't particularly well sequenced. All right, so just very briefly then, as I said, this is currently employed at JBC. We're now working on a completely de novo approach. So what do you do when you don't have a reference genome? And that could be very useful in the context of sort of an early warning system. And also incorporating information across multiple samples to get some more certainty and perhaps filter out those false positives. As I said, everything I've talked about is in the context of Illumina. Is a good question is whether this would work for nanopore, and it probably would, because we've got those adjustable error rates on the amplicons, but yeah, it's not something that we've tried yet. And yeah, and this is acknowledgements to the entire team of people that have been involved in the sort of bioinformatics side of the wastewater analysis. So yeah, you've got the whole group of people at DHSC, Hubert and Jazz in particular. And then Seb Bragadou at the Earlham actually developed this pipeline. I worked on the stats, and Paul Blackwell was, yeah, very, very useful in terms of discussing those and the test samples, obviously, from Liverpool and Sheffield, plus, yeah, lots of other people. So yeah, I'll leave it there.
Thank you, Chris.
How am I doing for time? Oh yeah,
you're good. You've got 5 minutes to your meeting, so all good. So this is really looking very promising, and it'd be good to have some questions. If we do have time for a few questions for Chris, so please do post some questions in your chat. They can be more general questions about wastewater, if you like. So Chris, what is at the moment, what is the kind of lag between, you know, a patient with a particular variant and then the kind of detection in wastewater, and what are those, you know, kind of what is that delay, and how is that delay broken down? Do you know, like you talked about the idea of it being an early warning system, which I think is quite exciting, but do you think that is practical?
This would be a question which would be much better for the experts at JBC, like Jazz cribbly in particular, but anecdotally, they see them very soon. If in some instances before they're observed in the genome sequencing.
Okay.
And also, what's surprising, unless again this is anecdotally, is you don't need a lot of individuals with a particular lineage to see it in the wastewater. So in some particular instances, I believe that they have seen a new variant in the wastewater before it was spotted with testing, and then they've been able to track down the individual with that with that particular variant.
That was going to be a question. So do you have a sense from, can your algorithm somehow estimate, or have you got a sense from the analysis that you've done of kind of how many individuals contribute to the signal? So if you take a particular wastewater catchment, is that, are you detecting the virus from one or two individuals, or is it kind of hundreds or thousands of individuals? Have you got any sense of that?
Yeah, no, that's a good question, and it's one that's outside of my remit. Okay, so if you think about it, what can we do from the DNA? The most we can say is how many different lineages of COVID are there, and their proportions. We can't really say anything directly from the DNA about the population. Instead, what they're doing is looking at other markers to answer that question. So there are various chemical markers, which and there's groups in the same overall project working on this. You can use chemical markers, oftentimes actually drugs. So things like caffeine is a good indicator of the population size. Caffeine, is always in wastewater, and it has these really interesting diurnal cycles. So, there's loads in the morning and it tails off through the day. And you can use concentration of caffeine if you can take account of degradation rates, etc., to estimate the overall population. Also, anti-epilepsy drugs. Any drug that doesn't break down quickly and is taken by a significant fraction of the population, the concentration of it can give you an indication of the population size. So there are people working on that.
That's
basically.
Interesting. I didn't know that.
No, it is interesting. I mean, it's been a really interesting experience for me. So it's it's an exciting overall project actually, and there's lots of people doing lots of interesting things on it.
No, very good. Thank you very much and it's good to see the kind of update from last time you spoke. That's really good. I know you've got to go to another meeting, so it's 3. I'll let you go off.
All right.
I'll let you know if there are any follow-up questions of Chris. Pop them in the chat and I'll get them over to him and get you the reply back. But thanks very much for coming.
So Nabil is worrying about what happens to the fish. Yeah, that isn't actually that's not a totally ridiculous question. I mean, the one they most worry about with the fish is actually estrogen. So there's a huge amount of estrogen in wastewater from contraceptive pills, and there is an idea that that's feminizing the fish. But yeah, whether the caffeine is having a big impact on the fish, who knows?
All the COVID.
That's actually quite an interesting idea. Anyway, all right. It's nice. Yeah, nice to see you. Bye then.
All right, good to see you. Bye for now, Chris. Bye.